Chapter 16, Simple Disease Risk Statistics. In this chapter, I want to explain a little bit about how you quantify risk, how we use it in genetics, and how you can read the report in terms of disease risk. So this is the simple part. This is really something that it would be good for you to read so you understand the report. There is another more advanced section where you find out how to multiply several disease risk statistics and so on. So the other stuff is really just for people who are interested and want to know more. And this is the simple stuff about how to read the reports. So first, there is a term that you're going to have to learn. The term is called odds ratio, which means kind of relative risk. And let me explain what it means. As I've explained in the chapter about genetics, the human body has the chromosomes, and the genetic code is part of the chromosomes. And then we have the genes, which are certain areas in this genetic code. And then we look at specific variations. So we don't look at the whole gene, because that's the same for virtually all people. But we look at the small variations where we know there is sometimes a different genetic letter from one person to the next. And then we know that a person, for example, for the diabetes gene, G in this position would mean a functional gene. We call this wild type. And A would be the mutated gene, the gene that doesn't perform its function. And this is what we call mutant. So really, if you look at a gene, at a genetic variation, you can have two different letters in this case. So, as I've explained in how genes are inherited, that's another chapter that I would recommend you to look at if you haven't done so already. Everyone has two copies of each gene. So one from your father, one from your mother. There's some examples, but for most genes this is correct. Some exceptions. Then a genetic variation can therefore be only in one gene, only in the other, none at all, or in both genes. So you get three different possibilities, both broken, both functional, or one of them functional. So with this example, G would be a functional gene, A would be a mutated gene. And the possible outcome is G on both genes, we call it wild type, so it's a G here and G here. G on one, A on the other, and A on both. So A on both, A, G would be heterozygous, is what we call it. So wild type, heterozygous, only one of the two genes, and homozygous mutant. These are the three possible outcomes. You might have recognized it could be AG, but it could also be GA. So it's not this gene that's broken, but it's the other one. But really in genetics we don't differentiate. So really having one of them broken is already heterozygous, and you don't differentiate. It's also not so easy to test which one of the two is the problem. So if you look at the GG wild type, as I said, both genes are functional, and here we come across this word odds ratio. The odds ratio is 1. So this is a gene that is associated with a disease. However, the gene is not broken, and the odds ratio is 1. So this person has a one-fold risk of developing the disease. The normal risk, the general population risk, so to speak. Now, if one of the two is broken, and this differs from gene to gene, but in this case it's called a heterozygous with an odds ratio of, for example, 2. This means this person has a two-fold risk. This is a one-fold risk, this is a two-fold risk, so double the risk of developing the disease. So if it is heart attack, this person has a normal risk of developing heart attack, and this person has a twice as high risk to develop it. So double the risk, because there's an odds ratio of 2. And then if we say it's 2 mutations, it's homozygous mutant, then you have an odds ratio of 4. So it's a 4-fold risk, or quadruple risk. So the odds ratio is really a relative risk of how likely it is that you develop a disease. A person with 2 A's will have a 4 times higher risk of developing the disease than with the GG. And as I said, it's relative. So it's really compared to a person with optimal genes, you have a 4 times higher risk. Compared to a person with optimal genes, you have a 2 times higher risk. So this is how odds ratios work. The higher the number, the higher your risk, and 1 is normal risk. And this is actually an extract from one of the reports. This is for diabetes, for example. Here are the genes that we test for. These are short names, TCF7L2 and so on. Then a genetic variation has a specific, it's kind of like a catalog number. And this is the number here. It starts with RS and then there's a number. This is a unique identifier that tells you this is exactly this genetic variation. Another geneticist will be able to use this information to check up on it. Then polymorphism, this is really just some scientific jargon. It means the A letter is changed to a G through this variation. A G is changed to a C at position 174, and the pro amino acid on position 12 is changed to ALA. So really, a geneticist will know what this means. Anyone else doesn't really need to know because this is just some scientific information. Now, genotype, that's actually interesting. This is the actual laboratory result. So, as I said, you have 2 copies of each gene. We look at specific genetic variation in the TCF7L2 gene, for example. And at this variation, it can be a C or a T. So that's the 2 different letters that could be there. And this person, as you can see here, genotype CC, has 2 Cs, so 1 on each. And that's the genetic result. This person has a AA on the other one, AA, and actually he is not heterozygous on any, otherwise it would be CG or TA and so on. So this is really the unique genetic result that the person has. And here's the odds ratio. So the CC genotype, the genetic result, the genotype it's called, has an odds ratio of 1. So if in this gene you have a CC, then you have a normal risk of developing the disease, diabetes. So it's not a risk. So if we look at this one here, for example, KCNJ11, if you have TT, so both genes here have the variation and the genetic defect, so to speak, then the odds ratio is higher, and it's a 1.65. So whilst people with CC have a 1-fold risk of developing disease, the person with TT has a 1.65-fold risk. Now this means 1.65 times likelihood of developing the disease, or 65% higher risk than people with the optimal genes. So if you're interested, you can work with this. To be honest, it's really some scientific stuff that is not of much relevance, because we use this kind of information to find out the risk and then decide what sort of preventive actions should be taken. It's really for people that are interested in it, but it's not necessarily important. Now there's an interesting one here. This is a CC genotype for this gene, and this person has a 0.91-fold risk. So it's actually lower than 1, which means it's a protection. So while he might have a 1-fold risk, this person has a 0.91-fold risk, so it's a 9% lower risk than a person with the other genes. So really this can be higher or lower. It might be a risk or it might be protection from a disease. Then here you see responder. If it says responder here, this means that this gene responds to something or makes you respond to something specifically well. In this case, it's a certain treatment. Certain medication works better with some genes and less good with others. So really this means this gene is a responder. It's this gene as well. It helps you. You will find this somewhere else in the report, what it makes you respond to better. Then, as I said, this is protective, so it's actually a lower risk than the normal risk, so it's protection. And these two create a risk, that's why it says risk here. And this is the genetic table. This is about as technical as it gets in the report. Again, this is just a lab result. You don't really need this kind of information to work with the program, but it's good that if you understand it. All right. So this here is the actual science behind it. This again is for experts that say, I want to know what's the scientific basis of this claim that we make. And this is just one of the genes, and you find them in the reports in this way. It will give you the name of the gene, some short explanation. It's really just for experts. And then you see the 3 possibilities. as I said, 2 Cs or 2 Ts, or 1 C and 1 T. That's the 3 options that we have. And here you see that's the result. So this person has TT. Now this percentage here tells you how many percent of the general population also have this genetic variation. So this person has a TT, and 8% of the general population also have a TT in this gene. This just shows you how common it is. Is it something that's very rare? Is it something that most people have, or how common is it? So it's just a comparison. So against all likelihood, this person has the rarest gene combination out of the 3. And here you see the meaning, the meaning that this, or the genetic traits that this genetic variation causes. For example, the CC causes no increased risk of diabetes mellitus type 2, and the drug metformin is effective. So really no risk, and also a certain drug works better. If you have a CT, you have a higher risk, an odds ratio of 1.23, which means 1.23-fold risk, 23% higher risk. And the drug metformin is less effective than normal. So metformin is not so effective for this person. You might want to use another drug. And here with 2 Ts, the risk is higher, 1.65. The drug metformin is also less effective. And in case you're interested, this here is the scientists behind it. So there are scientific publications that we base all of our claims on, and we state them. This is standard. A standard clean way of doing science to show what you base your claims on. So if you're interested and you understand the scientific jargon, then you can read up on it. And this is the kind of genetic information that you will find in the reports, and that will allow doctors or scientists to work with the information, check it themselves if it's really true what we're saying, and so on. Okay, so an odds ratio is the likelihood of developing a disease. Having a high odds ratio is bad, having a low odds ratio is good for your health in this case. Now, however, you need to be careful. A high odds ratio does not always mean it's a big problem. Let's say there's a disease that 20% of people get. And then you have a 1-fold risk of developing this disease, which means you have a 20% risk of developing it. If you have an odds ratio of 3, that would mean you have a 3 times higher risk than the person with the optimal genetic variations. So the calculation is a bit more complicated. If you're interested, look at the next, at the advanced disease statistics training. But simplified, odds ratio of 1 is 20% likelihood of developing the disease. Odds ratio of 3 is 3 times that. So it's 60% likelihood of developing the disease. So really an odds ratio of 3 for this disease is quite severe. You have a very high chance of developing it. And then you should take preventive measures to try and stop that. Now, let's look at this example. There is a fictitious disease called GMO syndrome. And it's a very rare disease. There's just one gene variant that exists that causes this disease. And the general population has a normal risk of odds ratio of 1. So a 1-fold risk of developing this rare disease, which is very low. And only 1 out of 100,000 suffers from the disease. So every person has a 0.001% lifetime risk. So a very low risk of developing the disease. Now, the odds ratio relative to the general population is 1, as I said, because the general population has a 1 times risk of developing the disease. Now let's take a woman. She is unlucky. She has a genetic variation that causes an odds ratio of 60. So it's a 60 times higher risk to develop the disease compared to all other people. So this sounds very severe. However, if you look at the numbers, the average has a 0.01% lifetime risk, an odds ratio of 1. If you now look at what is the odds ratio for 60, that's kind of like similar to calculating 0.001 times 60 is 0.06%. So even though she has an odds ratio of 60, which sounds very severe and dangerous, it's still a very low risk of developing the disease. And this means that this kind of genetic test could give a result of 60-fold risk. However, it would not be relevant for the person's health. So really, having a high odds ratio is not good enough. You always need to look at how common is the disease. If you multiply the disease by the likelihood of disease by 60, is it then even relevant or not? And this is what we do. We look at the genetic effect on the disease risk, and we filter out the right kind of genetic variations that do make sense. However, some other companies, some other laboratories, usually internet genetic testing, they don't filter this out. They just say this variation causes 60-fold risk, and it's communicated. The person can sleep even though it's completely irrelevant to health. So this is an important thing to consider and something that we do for you. Now let's look at a few different examples of where a genetic test makes sense and where it doesn't. For example, let's look at the disease that occurs in 5% of the population, and the genetic variation increases the risk to an odds ratio of 7. So 5% of the population get the disease. Any person's risk is 5% of developing this disease. Now, one person has a genetic variation and a 7-fold risk, so 7 times higher than anybody else. And the probability is increased from 5% to 30%. Now, you might calculate 5 times 7 is not 30. This is because the calculation is a bit more complicated, but really the risk or the probability of developing increases to 30%, and if there are preventive measures, it might make sense to do those if you have a high risk, because you might get the disease. So this is a relevant kind of genetic test that you could do. Then another example, the disease occurs in 1% of the population. The genetic variation increases the risk to an odds ratio of 1.1. Now what's going to happen here? You increase from 1% to 1.1% risk. Really, there's no benefit in knowing you have this genetic variation. The risk is negligible. So this kind of genetic test doesn't make any sense. Then the disease occurs in approximately 1%. The genetic variation increases the risk to odds ratio 60. Now, that's interesting. Even though the disease is relatively rare, the risk is quite high. And it increases, again, it's not really 60%, 1 times 60, but it's a bit different, but it's from 1 to 45%. So again, from hardly a chance to develop it, it's a 50-50 chance that you will get it. And if there's prevention, it would be a good idea to follow this quite strictly to reduce your risk again. So this is a relevant kind of genetic information. The disease occurs in 0.001%. The genetic variation increases the risk to odds ratio 90. Again, from 0.01 to 0.06, it's not relevant. The disease occurs in approximately 0.001%, so extremely rare. So you will already guess that's not going to be a sensible test. But mutations lead to developing the disease in all cases. So 100% disease risk caused by the genetic variation. This is, there are some diseases where this is the case, and this of course is relevant because the likelihood of developing it is 100%. And this is usually the case for rare genetic disorders that have a very, very strong impact on health. So this is a highly relevant genetic test. So really there's some things to consider, and a laboratory that offers genetic tests that increase the risk to 0.06 and communicate it as a high risk event should not be taken seriously. So really make sure that the science is shown, that the disease is common enough, and so on. So as I said, this kind of selection has been done by our experts in our panels, so you don't need to worry about this there, but it's something to keep an eye on. Now, since we can find out the genetic risk, why don't we just say your risk of developing the disease is something %? Wouldn't that be more interesting? Isn't it doesn't it tell me more to find out I have a 80% risk of developing, likelihood of developing Alzheimer's disease? Isn't that better than knowing my risk is 4 times higher? And for this, I want to show you an example of why we don't do this usually. This person has an odds ratio of 3 through genetic variation of developing Alzheimer's disease, dementia. And the disease probability is on average 20%. 20% of the general population develops Alzheimer's disease. Now, this is the kind of information that a genetic laboratory would communicate. But why not say 20% times 3? Because there is a prevention. Although Alzheimer's disease, when it has occurred, is difficult or impossible to treat so far, there are some environmental factors that help you to either delay the onset or reduce the likelihood of it to develop. And scientific studies have shown a number of these factors that have been beneficial. For example, drinking 2 to 5 cups of coffee per day reduces the risk to 36%. So more than 60% lower risk of developing Alzheimer's disease if you drink coffee, which is surprising. The reason being coffee contains a lot of antioxidants. It's a very strong antioxidant solution. And Alzheimer's disease is associated with oxidative stress, free radical damage in the brain. And this is why people think, or scientists think, that this protective effect occurs. But even though the genes say you have a higher risk, if the coffee consumption says you have a much lower risk, and then you also do exercise, regular exercise, also lowers the risk, then by having high risk here, but lower and lower risk here through lifestyle changes, you can actually reduce your risk. This is a statistical approximation, of course. This is not an exact number, but this would, in the end, reduce your risk, even though you started off with a 3 times high genetic risk, to around 13%. So really, you have a high risk, you have other ways of lowering your overall risk, and this is why it is difficult to say your disease probability is a certain percentage, because the aim really is to reduce this. The same person living an unhealthy lifestyle, smoking, drinking, no intellectual challenges, will increase his or her risk of developing the disease. So here the probability can be even higher. So while a laboratory can really detect the 3 times genetic risk, what else the person does with his risk is up to him. So really it's difficult for a laboratory to calculate this is your probability of developing the disease, because it is modifiable. It changes with the lifestyle. So really what a laboratory needs to do is tell you the genetic risk and tell you which way your lifestyle should be changed to counteract the genetic risk. That's the concept of preventive genetics. So the genetic risk is stable and unchangeable. It is inherited and it will stay the same. Every other laboratory analyzing the same genes should get the same results if they work scientifically. So different analyses of the same genes should yield the same odds ratio, even when conducted by different laboratories. The disease probability is affected by positive or negative environmental influences, so you can increase it even more or reduce the risk. And thus the probability of disease cannot be reliably measured in a laboratory, because we don't know, does the person drink alcohol or does he drink coffee when he has an Alzheimer's disease risk. And really the aim of a preventive genetic analysis is not to predict the probability of developing a disease, because that's just diagnostic. There's no use in this information, but is to influence the risk of developing the disease by modifying the lifestyle or the environmental factors in such a way that the disease risk is lowered even though there's a high genetic risk. That's the aim. So really, that is why we communicate your relative risk. It's a stable number that is there in your genes, and then we try, or preventive genetics tries to help you modify your lifestyle to prevent getting this disease. And that's the end of chapter number 16, the simple disease risk statistics. And for people who want to know more, only for those, it's not necessary. You can also look at the next chapter about advanced disease risk statistics, and you're going to find out more information of how odds ratios are combined and so on. Thank you.